Hi everyone, in this video, we will learn inlet and exit gas analysis during a fermentation. Inlet and exit gas analysis, measurement and recording of the inlet and exit gas composition is important in a fermentation. Now, what do I mean by inlet gas and exit gas? This is a picture of a fermenter, and the inlet gas is the gas that you are introducing via the sparger. So the sterile air that is introduced via this sparger in the fermenter is the inlet gas, and the exit gas is the exhaust gas that leaves the fermenter from the top. Now, yes, you will measure the inlet and exit gas. You will record the inlet and exit gas composition. That is the gas which you are introducing via the sparger and the exhaust gas. But which gases will you monitor in this inlet gas and the exhaust gas? We are talking about oxygen and carbon dioxide. By observing the concentrations of carbon dioxide and oxygen in entry and exit gases in the fermenter and knowing the gas flow rate, it is possible to determine the oxygen uptake of the system. You are giving the oxygen via the sparger. You are providing the air to microorganisms. But whether the oxygen is getting dissolved or no, whether they are utilizing that oxygen or no. So what is the oxygen uptake of your system or the fermentation? You can get the idea of that. You can also find out if the organisms are utilizing oxygen, are they producing carbon dioxide? So the carbon dioxide evolution rate and the respiration rate of the microbial culture. Obviously, you would have more oxygen in the inlet gas, and in the exhaust gas, you would have less oxygen and more of carbon dioxide. How would you monitor the oxygen and carbon dioxide in inlet and exit gases? Let's see that. So first we will see how would you monitor the oxygen concentration in an inlet gas or an exhaust gas. So oxygen concentration can be determined in an inlet or exit gas by a paramagnetic gas analyzer. This is the sensor that will help you in determining the oxygen concentration in inlet and exit gas. Oxygen has a strong affinity for magnetic field. It shows paramagnetic behavior, and this property is exploited in paramagnetic gas analyzer. There are two types of paramagnetic gas analyzers, deflection and thermal type. We'll be seeing the principle of both of these in detail. Let's see first the deflection type paramagnetic oxygen analyzer. Now, this deflection type paramagnetic oxygen analyzer, it's a very popular sensor. It predominates, and this paramagnetic oxygen sensor consists of a cylindrical shaped container inside of which is placed a small glass dumbbell. You can see this dumbbell test body. So in the cylindrical container, there is a small glass dumbbell. Now this dumbbell, what you can see here, is filled with an inert gas such as nitrogen, and this dumbbell is suspended on a platinum wire, which you can see here. Okay, here it's written quartz, but it can be of a platinum wire as well. And this dumbbell is within a non-uniform magnetic field. You can see a magnet here. You can see the North Pole and South Pole. So yes, there is a small glass dumbbell. The dumbbell is filled with an inert gas such as nitrogen, suspended on a platinum wire within a magnetic field. And this dumbbell is designed to move freely as it is suspended from the wire. It is free to rotate about an axis. Now you are using this sensor to determine the oxygen concentration in your inlet gas or exhaust gas. When your sample containing oxygen is passed through this sensor, and as we have already told that oxygen shows a paramagnetic behavior or displays a paramagnetic behavior, it has a strong affinity for magnetic field. So when you introduce your sample, the oxygen in your sample will get attracted to the stronger of the two magnetic fields, and this will cause displacement of this dumbbell. Okay, and in short, I would say this dumbbell would rotate. It would move away from its original position. A precision optical system, which you can see in the lower part of the diagram, consisting of a light source, a photodiode, and an amplifier circuit, is used to measure the degree of rotation of the dumbbell. The top view is just, this is actually the side view. It's one of the diagrams. And you can see the top view here, and you can see the light dividing mirror, a photo tube, a lamp, lens. So this optical system is used to measure the degree of rotation of the dumbbell, how much the dumbbell has rotated because of oxygen. An opposing current is applied to restore the dumbbell to its normal position, and this current required to bring back the dumbbell to its normal position is directly proportional to the partial pressure of oxygen, and it is represented electronically in percent oxygen. Okay, summary, this is a sensor that will tell you the oxygen concentration in your inlet or exhaust gas, and it exploits the property of oxygen, that oxygen displays a paramagnetic behavior and has a strong affinity for magnetic field. So when you introduce oxygen, the oxygen would get attracted to this magnetic field, and since the oxygen got attracted to this magnetic field, this will cause the rotation of this dumbbell, as the dumbbell is free to rotate, and the force applied or the current required to bring back the dumbbell to its normal position will give you the oxygen concentration. Okay, so since the dumbbell can rotate, this is called a deflection type paramagnetic oxygen analyzer. The second type of paramagnetic oxygen analyzer is the thermal type paramagnetic analyzer. And in this, a flow through ring element is the detector component. You can see there's a ring here, and the oxygen sample, or the sample in which you are suspecting the oxygen, is allowed to come through here. And again, since this is a paramagnetic oxygen analyzer, this will also exploit the same property of oxygen, that it has high affinity towards magnetic field. And there is a magnet in the central tube. You can see there's a ring, but there is a central tube as well, and there's a magnet in the central tube. So when the oxygen will be introduced, or the sample would be introduced from here, obviously the sample would be attracted, or the oxygen in your sample would be attracted towards this magnet or magnetic field, and the oxygen will come to this central tube. So the paramagnetic oxygen content of the sample is attracted to this magnetic field in the central glass tube, where the oxygen will come. And the magnet is not the only thing here in the central tube or the magnetic field. There are resistors as well. And these resistors heat the gases. These resistors are connected to a Wheatstone bridge circuit to detect variations in resistance due to flow rate changes. Now the oxygen in your sample got attracted to the magnetic field, and it has come to the central tube. And the resistors in the central tube will heat the oxygen. And the oxygen in the heated sample, or I can say the heated oxygen, loses high proportion of its paramagnetism. Yes, the oxygen which will get heated now will lose high proportion of its paramagnetism, and now it's no longer attracted to the magnetic field. So the heated oxygen will come through, you can see these arrows here, will come out of the central tube. The heated oxygen will come out of this central tube and will be displaced like this. Now, but the sample is continuously passed through here. So the cool oxygen in the incoming gas will now be attracted. So this cool oxygen in the incoming gas would be still attracted to the magnetic field. And when the cool oxygen in the incoming gas will be attracted to this magnetic field, again the resistors will heat that oxygen. After getting heated, it will lose its paramagnetism, and the heated oxygen will leave from here, the red circles that you can see. But the cool oxygen will now be attracted and it will displace the hot oxygen. This displacement action produces a convection current. The flow rate of this convection current is a function of the oxygen concentration and can be detected by resistors. The resulting gas flow cools the winding A. The winding A, which you can see here, the winding A is this side, and since the cool oxygen is coming, the resulting gas flow cools the winding A, and this side is the winding B, and this heats the winding B, or the winding B gets heated as the heated oxygen is leaving from that side, and this resulting temperature difference imbalances the Wheatstone bridge. And since the oxygen is heated air, this type of paramagnetic oxygen analyzer is called as thermal type. So both these analyzers are paramagnetic oxygen analyzers, and both can be used to determine the oxygen concentration in inlet or exhaust gas. One is deflection type, another is thermal type, and both, based on the same principle, although you can see the details are different, but the main principle is same, that oxygen has affinity towards magnetic field. And these oxygen analyzers, when you are determining the oxygen concentration, one more thing you have to pay attention to is that it would be necessary to monitor and record the atmospheric pressure if oxygen concentrations in inlet and exhaust gas are to be determined using these oxygen gas analyzers. Why pressure? Because these paramagnetic analyzers are susceptible to changes in barometric pressure, and a change of 1% in pressure may cause a 1% change in the oxygen concentration. This size of error may be very significant in a vessel where the oxygen consumption rate is very low and there is a very little difference between the inlet and exit gas composition. So pressure changes should be constantly monitored when you are determining the oxygen concentration via this paramagnetic analyzers. So the pressure can be monitored using the pressure gauges or pressure sensors, which we have learned in one of the previous recordings. So this was if you want to monitor the oxygen in inlet and exhaust gas. So paramagnetic gas analyzers, deflection and thermal. Now, how would you monitor carbon dioxide in inlet and exhaust gas? For carbon dioxide, the sensor is different. You have to use a infrared sensor to monitor the carbon dioxide in inlet and exhaust gas. This infrared sensor has an infrared source, which you can see here. That means a light source. Now, how you have a tungsten bulb in a colorimeter or a hydrogen or deuterium bulb along with a tungsten bulb in a UV visible spectrophotometer? So there is an infrared source in this infrared analyzer, and then you can see there's a chopper. This line, what you can see, there is a chopper, and this chopper is driven by a motor. This chopper ensures that energy passes through each side of the optical system. There is a sample cell in which your air sample is there. There's a comparison or the reference cell, and there is a detector. This detector is sensitized at a wavelength at which the gas of interest absorbs infrared energy. In this case, the detector will be filled with carbon dioxide. So it's a positive filtering method, infrared analysis. Now, what happens actually here is, when you have your gas or your sample may be an inlet gas that you are introducing via sparger, or your sample is an exhaust gas, whatever is your sample is filled here. And when you keep your sample in the sample cell in the path of this infrared source, and when the infrared source will pass through here, the carbon dioxide in your sample will absorb the infrared light. The carbon dioxide in your sample will absorb the infrared light, and if the infrared radiation is absorbed by the carbon dioxide in your sample, obviously the transmitted radiation will be of reduced energy. So the optical system or this detector will sense this, that the radiation coming out of the sample is of reduced energy, and that happens because the carbon dioxide in your sample has absorbed the infrared radiation, and that's why less radiation is reaching the detector. How much radiation would be absorbed, that depends on the concentration of CO2 in the sample. There's an amplifier, there's a readout. So This will tell you the concentration of carbon dioxide in your inlet and exhaust gas. They say it's expensive to have a separate carbon dioxide and oxygen analyzer for each separate fermenter. Let's say in an industry, if there are 5 fermenters, to keep a deflection or thermal type paramagnetic gas analyzer and to keep this infrared analyzer for each fermenter would be very expensive. So it may be possible to couple up a group of fermenters via multiplexer to a single pair of gas analyzers. If you have 5 fermenters, all of them can be coupled via multiplexer to a single pair of gas analyzer, the paramagnetic oxygen analyzer for oxygen, which may be of deflection or thermal type, and this infrared analyzer for CO2. Gas analysis readings can be taken in rotation for each fermenter every 30 to 60 minutes. And in many cases, this will be adequate. Alternatively, the gas analyzers can be replaced by a mass spectrometer, which can analyze number of components along with oxygen and carbon dioxide. So this video was all about how would you monitor oxygen and carbon dioxide in inlet and exhaust gas of a ferment.